Our project concerns dynamic imaging of the immune system and an application of machine learning as a data analysis strategy. Specifically, we aim to track populations of T cells, shown here in red, from hundreds of gigabytes of raw voxel data in multiple fluorescent channels. We began by running a 3D segmentation algorithm to create 2D surfaces around potential T cells. Principal component analysis on this data showed that the majority of T cells form a distinct cluster. However, there are also many spectral outliers owing to overlap with other fluorescent structures within the lymph node. To engineer features to identify these outliers, we computed intensity-weighted centers of mass for the 6 different spectral channels and took pairwise differences. These features capture the scenario when 2 different cells in different colors are overlapping within a given surface. We also created a series of features to capture heterogeneity induced by the imaging system itself. Shown here is a fluorescence excitation correction based on how much light was scattered before exciting the fluorescent cells.
We perform regularization and variable selection via the elastic net procedure. In particular, we performed 100 bootstrap resampling procedures from both T cell and non-T cell data of approximately equal sample size. The figure on the right shows the 15 predictors picked by the elastic net. So for example, the bounding box AA length 2 was picked 100 times, etc. Within each bootstrap sample set, we further ran 1,000 iterations of randomly picking training test set partitions. This step was performed in order to assess the stability and overall distribution of the lambda parameter. In the first figure on the far left, we can see the distribution of the lambda parameter. The next graph represents the cross-validation error and misclassification error for each bootstrap partition. Utilize a pull-based sampling active learning procedure, so the entire data is evaluated and ranked before selecting the best query. Instances are evaluated in a greedy fashion according to an uncertainly sampling query strategy. More specifically, we use label propagation, a common graph-based, semi-supervised learning algorithm. The key assumption is that instances occupying the same manifold are likely to share the same semantic label. With W being the affinity matrix, F the labeling scores, and Y the initial labels, the final labeling scores are defined as a solution that minimizes the cost function presented here. Intuitively, each point receives information from its neighbors, which is the first term, and also retains the initial information, which is the second term. The label of Hx is then just the argmax of a corresponding column. Once labels are assigned, we compute the entropies of the transducted label distributions. Our samples with the highest entropy are then picked, and their labels are revealed. The procedure gets repeated with the sample set of known labels, now increased by odd. We initialize the algorithm with 10 labeled samples, further adding 5 per each iteration. Final confusion matrix is shown in the current slide. Additionally, we tested 2 different query strategies, entropy-based and random sampling. While both strategies performed well, entropy-based demonstrated slightly higher and more stable accuracy on unlabeled samples.